And I want to start by saying that this study that I will describe is a true, trans-Columbia, trans-campus, inter-campus collaboration that involves people from the Columbia Biobank, Jim Goldman and Peter Canol, people from the basics, from the main campus. Stuart Firestein helped us with the studies. And but the three key figures that I want to really thank is a postdoc in my lab, Mariana Zazicka, a graduate student, Albana Kodra, and an amazing physician who joined Columbia just before the pandemic, and he had interest on olfaction and reached out to us and spearhead the experiments that I will talk about. And speaking of Mariana, I just want to say that when our paper came out, it was published almost the same day that the war in her motherland started. So I want to give a shout out to her and her family. And I have to say that seeing her working on this project makes me realize why Ukraine is not losing this war. So before I talk about olfactory deficits in COVID patients, I need to give a little bit of an introduction in the olfactory system so you understand the basic biology, which will be very helpful for understanding why COVID hijacks this sensory system. So olfaction is the sensory system that detects volatile chemicals. And this detection is accomplished by G protein coupled receptors, the olfactory receptors, which are expressed almost exclusively in olfactory neurons. So the olfactory receptors sit at the cilia of the olfactory neurons, and as the volatile chemicals enter the lumen of the olfactory epithelium as we breathe air, they become activated by specific interactions with these volatile chemicals and initiate the signaling cascade that then is transmitted to the olfactory bulb and the rest of the brain. Now, the key feature of this sensory system, which is very different from any other sensory system, is that it detects an astronomical number of chemicals, and it does so by basically committing about 5% of our genes in odor detection. So the olfactory receptor genes, the genes that encode for these receptors, can be found in every chromosome, really. And in the mouse, there are 1,400 of them. In human, there are around 1,000, but most of them are pseudogenes. There's only about 300 to 400 functional olfactory receptors. And in every chromosome, they are not alone in general. There are a few exceptions, but they're organized in large genomic clusters that can be as many as 200 olfactory receptor genes in a row. And a key feature of this system for olfaction to work, like the immune system, a key requirement is that every cell expresses one of these receptors. So there is a stochastic mechanism that really uses enhancers that exist in every one of these clusters. These enhancer sequences are pretty much shared between every cluster. They are recognized by three common proteins, LHX2, EBF, and LDB1. And the commonality of this problem, of this regulatory system, raises a question of how do you activate only one of these genes, right? So if you have 60 clusters with 60 enhancers regulated by the same factors, you could imagine a situation where each one of these enhancers loops in and activates a local gene, and that would result in a system of co-expressing multiple olfactory receptors, which would be detrimental for olfactory perception. So years of work in my labs and others have shown that the solution for this problem is that these genes are not scattered randomly in the nucleus, but instead they are organized in 3 to 5 genomic compartments that have the size of the glomerulus. of a nucleolus, I'm sorry. And they are consisted of two different features. One is repressed heterochromatic olfactory receptors that are completely silent. And in one of these compartments, there is an enhancer complex consisted of the individual enhancers of all these clusters that come together. And this enhancer complex associates with one of the olfactory receptors, which is randomly selected and transcribed at very high levels. And I just want to show you some of the images that led to this model. Here is a DNA fish experiment in an olfactory neuron with a probe that detects all 1,400 olfactory receptor genes. And you can see that instead of being scattered in the nucleus, they're organized in 4 specific compartments. And here's a similar experiment where we do a DNA fish for these enhancer sequences, and we are monitoring the active olfactory receptor. And we often see a situation where the active allele, that is the one that is transcribed, is surrounded in a cloud of enhancers, which can vary in size from cell to cell. So I'm telling you all that because, and I need to add one more key information. There is a feedback process in this system. So when one olfactory receptor is chosen for transcription, is then translated in the endoplasmic reticulum, and that somehow initiates, activates a kinase called Perk, which it's a kinase that resides in the ER. And Perk affects global translation in these neurons that results in the translation of a transcription factor that is involved in stabilizing the silencing of the genes that were not chosen. So basically, there's a spatial mechanism that allows one olfactory receptor to be transcribed at a time. And then there's a system in place that detects the product of this receptor and tells the nucleus stop picking up new receptors, stick to that one. So this is basically the background you need to follow this talk. This is what my lab studies, and early on in the pandemic, you know, anecdotal news first, and then studies with high numbers of people start emerging, showing that one of the early symptoms of SARS-CoV-2 infection is the loss of smell. For many people, is a complete loss of smell that is happening suddenly, and in most cases recovers equally suddenly. So we were intrigued by that because when we start learning about the mechanism by which the virus infects cells, we immediately start asking whether olfactory neurons, which are the neurons that detect odors, express the host receptor proteins that are required for viral infection. And based on our own data and a very rigorous study that was published early on by Bob Dadach and I and many other labs participated in that, it became apparent that olfactory sensory neurons, the cells responsible for the detection of odors, do not express at all the host proteins. So this raises several concerning possibilities. One would be that the virus have different mechanisms of infecting the olfactory neurons. And if that's the case, we should figure that out as fast as possible. The other concerning probability was that the anosmia was not peripheral, but the virus propagates in the brain, possibly through the olfactory tract. And that was also something that we had to figure out as fast as possible. Jonathan over the West reached out and asked us if we would be interested in analyzing autopsies of patients that died at New York Presbyterian from COVID. It took us quite a while to to get the anatomy right. Human olfaction is very different from mouse olfaction. So there was a lot of trial and error in the beginning. And I have to say a lot of early studies that suggested that olfactory neurons were infected by the virus were looking at the wrong place. And I think it's important to first figure out where the olfactory epithelium actually is in humans. So we did a lot of work on that, and we use immunohistochemistry with markers that we know are markers specific for olfactory neurons. We also did some single cell RNA-seq studies to confirm that we are able to isolate regions of the nasal epithelium that are most enriched for olfactory neurons. And after we establish that, we start analyzing these samples. And the first thing that we observed to our relief was that olfactory neurons are by and large not infected by the virus. So we see a lot of signal at the area of the sustentacular cells, but very little signal colocalizing with markers for olfactory neurons. And luckily for us, a very comprehensive study by the Lomvardas group confirmed in an unequivocal way that this is true. So we didn't have to spend a lot of time quantifying and making sure that this is general from autopsy to autopsy. So we could move on into actual functional experiments to figure out why anosmia is happening. So we did what we know how to do. We collected this autopsy tissue and we started analyzing by bulk RNA-seq. And we had several infected samples. Controls were more difficult to find, so the numbers are not great for the controls. But the general pattern that, general trends that emerges from this analysis is that tissue from infected autopsies has in general much lower levels of olfactory receptors than the tissue from controls. And there was one control sample that didn't fit this pattern. So I asked Albana, who was analyzing this data, to look for other viruses that might be present in this genome. And it was very exciting to us that by blasting the RNA-seq data, she find that actually this patient was infected with a old-fashioned coronavirus, which is known to be causing olfactory deficits. Like we all know the common cold causes olfactory deficits, but we always thought that this is because of the physical obstruction of odor entering the olfactory epithelium through mucus. But it seems that this infection also affects the levels of olfactory receptors. And an important other finding is that a lot of other molecules that are involved in signaling or trafficking of olfactory receptors are significantly downregulated compared to the few control samples we had. So it seems that the infection coincides with a general damping of the ability of the neuron to detect odors and transmit that signal to the brain. Now, these samples were extremely variable, as one would expect. And I have to say this was the first time my lab did anything with human samples. And since then, I have gained tremendous respect for all of you who work with humans. I don't know how you put up with this variability, but it's pretty maddening for someone who's used to work with very control conditions. So if we do a PCA analysis of our RNA-seq samples, there is no clustering at all between infected and non-infected samples. But if we do the PCA analysis only using the 1,000 or so olfactory receptor mRNAs, there's a clear clustering between the infected and the control samples. And the only control that goes with the infected is the one that has the other coronavirus. And here is a summary of the levels of all the olfactory receptor genes between controls and infected. So that was the result that really start convincing me that what we are observing is a real, it might be a real effect of viral infection. To confirm that, we reached out to Benjamin Tenover. He was at Mount Sinai at the moment. Now he's at NYU. Ben is a virologist, immunologist who had established hamsters as a model for viral infection for numerous reasons. So we asked to collaborate with him and see if there is a causal effect of viral infection to the changes in olfactory receptors. So because of the model organs, we were able to do more elegant experiments than the bulk experiments we did in humans. We started with a time course experiment in hamsters that are either not infected 1 day, 3, and 10 days post-infections. And these experiments allowed us to quantify cellular changes. I forgot to mention that our bulk RNA-seq data clearly suggested that the downregulation of olfactory receptors and of olfactory signaling molecules is not due to the depletion of olfactory neurons. A lot of olfactory neuron markers are stable throughout the infection. And this was also confirmed by the single-cell RNA-seq experiments. But these experiments also show that there's especially at 3 days post-infection, there's a massive increase of microglia, T cells, B cells, macrophages in the olfactory epithelium that later goes down as the infection goes away. And there is also, according to this data, a significant reduction of the sustentacular cells, which are the cells that express the host proteins that the virus can infect. And these are the cells that our human data had suggested are directly infected. So here is the detection of the viral genome. We can see it mostly in microglia that has been recruited to the olfactory epithelium. And there is a little bit of signal in sustentacular cells and some of the stem cells of the olfactory epithelium, which may be important for understanding the long-term effects of COVID. And there is very few cells. I actually believe that this is the noise of the single-cell RNA-seq experiments. There are very few neurons that have the virus. And more importantly, the number of, even if this is real infection, the number of neurons that would be infected would not justify loss of smell. Now, importantly, we see the same reduction of olfactory receptor genes. It becomes significant 2 days post-infection and peaks at 4 days post-infection and slowly recovers. And again, this is not due to the loss of olfactory neurons. There is a little bit of reduction of markers of olfactory neurons, but not such that would explain this effect on olfactory receptor expression. And here's a quantification of the transcriptional effects by bulk RNA-seq, similar time course experiments. You can see at 4 days post-infection, there's basically these cells shut down the ability to detect odorants. Both olfactory receptors cumulatively are shut down, and a lot of the other accessory proteins that are required for the sense of smell. And at the same time, actually earlier than that, there is a massive upregulation of immune response genes, antivirals, inflammatories, and such that gradually starts decreasing as the infection disappears. So by 10 days post-infection, we don't detect the virus anymore in the hamster olfactory epithelium. And I want to show some, independent confirmation of this observation. Here is immunofluorescence for adenyl cyclase 3. The protein that generates cyclic AMP in response to odor detection. It's essential for olfaction. Here's a control hamster olfactory epithelium. Here's an infected epithelium. This is the few cells that have viral proteins, but a lot of cells that are not infected downregulate at the protein level this critical protein, and the same is true at the transcriptional level. So again, the general point is that we see a very strong transcriptional response in molecules that are essential for olfactory detection in a non-cell autonomous fashion. So the obvious question is, if you have 1,000 different genes, we know that there are some variations in the way that these genes are regulated from the dorsal to the ventral part of the olfactory epithelium. So what could be a general mechanism that assures that all or the majority of these genes are downregulated upon viral infection? So we went back to the what we know to do best, which is to study the architecture of olfactory neurons. For this type of experiments, we use the genomic-based method that called Hi-C, and Hi-C very briefly measures the frequency by two genomic loci are near each other by measuring how frequently you can force them to ligate to each other. And this is a sequence-based method that measures ligation events. And there is a long literature using this technology that has described how the genome becomes organized in the nucleus. So here I'm showing you a Hi-C experiment, and I'm plotting only olfactory receptor genes from all the hamster chromosomes. Basically, in the diagonal, we are plotting every single chromosome that contains olfactory receptors. These very dark squares basically are individual chromosomes, because these genomic interactions occur much more frequently within chromosomes than between chromosomes. But you can see over here with red is signal of contacts, for example, of an olfactory receptor on this chromosome with an olfactory receptor on this chromosome. So these are interchromosomal contacts that appear very frequently in hamsters, as we had described them appearing in humans. So this is the control tissue. 1 day post-infection.
Stavros, this is a 2-minute warning.
Okay. 1 day post-infection, there's there's an obvious downregulation, and that peaks at 3 days and does not fully recover by day 10. And this is the quantification. These are very significant changes. So I'll go back to the fact that these are non-cell autonomous effects, because very few olfactory neurons are infected. So Max Gottesman had this brilliant idea that he suggested to me when I was showing these results in the department, to take the serum from infected hamsters, remove the virus, and expose it to serum from naive hamsters and see what happens to the nuclear organization. So if we do that, we know that there is any viral infection going on. Here is a control of 1 day post-infection. And here is serum from mock infected hamsters, and here is serum from SARS-CoV-2 infected hamsters. You can see the effect it has in genomic organization. Again, pointing out that this is a non-cell autonomous effect. And we were hoping that we would use this strategy to fractionate and find the key molecule. We have not gone there yet, but this doesn't mean we have stopped trying. There is some transcriptional changes in olfactory receptors that follow these effects. And importantly, we see the same effect on infected humans compared to control samples. So to summarize, and we think that what happens in this system is we have this very well organized nuclear architecture that is essential for expression of olfactory receptors and a lot of their signaling components. When the virus infects cells that are infectable in the olfactory epithelium, there's some type of cytokine released that disrupts this structure and leads to anosmia. And I will skip my last slides, and I will go straight to the acknowledgements. Again, this special duo of postdoc and graduate student, Jonathan, has been critical for this experiment, Benjamin Tenover and his group, and of course, all the people from Columbia, Jim Goldman, Peter Canol, Max, and Stuart Firestein have been instrumental for these studies. And I will stop here and take questions.
Thank you very much, Stavros. And again, I encourage the audience, if you have questions, please raise your hand. Or if you need to, you can post them in the Q&A. And I would like to get started. Could you comment very quickly on the normal rate of protein turnover in the olfaction system in an uninfected individual? And does that change upon infection? Are you actually clearing out these proteins due to the infection itself?
Yeah, that's a very, very good question. We had done some experiments in the past. We have some TETO system where we can express an olfactory receptor and then shut off the expression of this gene and find out how long it takes for the proteins to be cleared. And we had found that it takes about 4 days for the receptor protein to become undetectable. So yes, I think that you're pointing to something very important, that maybe there is an accelerated mechanism of removing these proteins, and we are just scratching the transcriptional effects, but there might be some effects with protein turnovers that we have not thought of. Now, in regards of the anosmia, it is also possible that, you know, if you reduce a little bit, not only the receptor, but the G proteins and the L cyclase 3 and the channels, that it's possible that small reductions throughout the pathway are sufficient to reduce olfactory sensitivity. So it's possible that this contributes too, but what you suggested is very interesting, and I think we should try to follow up with that.
We have a question from Jian Shin. Jian, you should be able to unmute yourself and ask your question.
Hi, it was a great talk. I was wondering whether you see any changes of gene expression or protein expression of nuclear lamins or nuclear envelope proteins.
Yeah, that's a great question.
After infection, right?
Yeah,
because those proteins are like important for genome organization.
Yeah, so actually, we had published in the past that olfactory neurons obtain this genomic organization because they shut down the expression of lamin B receptor, which is a protein that normally holds the olfactory receptors at the nuclear periphery. And the first thing we checked was that if somehow the infection induces upregulation of lamin B receptor, but it doesn't. So it's not that. That would be great because we would have the answer, but it's not that, unfortunately.
Okay, good. Thank you.
Thank you.
Stavros, if I can ask one more question, this one's going to be bizarre. Maybe you'd laugh at me. When a cell becomes infected or experiences an infection, and you get these changes in nuclear organization that cause changes in expression, and it's also affecting the receptor gene itself, but the sense of smell comes back and it comes back the same. How does the cell know to reactivate the correct genes after infection is cleared?
That's a great question. It's not laughable at all. And I think it might be relevant to the few people that are having longer effects versus the ones that are recovering very, very fast. So there are two options, right? One is somehow you start disrupting this organization, and then when it comes back in a way that results in the activation of the same gene. And then the neurons would be functional and perception will be identical. If that doesn't happen and the recovery results in a random choice of any receptor available, that would mean that now you have a neuron that expresses the receptor for cherry, and it projects to the area of the brain that detects banana, right? And that not only it will be confusing, but also every other neuron will be making random connections. So that would lead to complete anosmia until these neurons die. There is some evidence that if you don't form this convergence of all the axons that detect the same odorant, and you don't have synchrony in their firing pattern, these neurons eventually die. So it's possible that this does happen, and the recovery for some patient takes longer because you need basically to wait for these neurons to die and for newer cells to replace them and create again a functional olfactory system. It's also possible that there is some type of histone modifications that are placed on the active receptor that allow that receptor to be chosen again, because this is essentially the only receptor that is not heterochromatic. So it's possible that after the structures recover, this is the one that will make contacts with other enhancers. But if you keep them apart for too long, maybe it loses these marks, and eventually the choice happens from scratch. So I was thinking that maybe, you know, timing of how fast you clear the virus and you allow these structures to come back together could be a determinant on how fast your olfactory dysfunction happens. And yeah, these are fascinating questions. Unfortunately, the only way to test all these things would be if we do experiments in a mouse model where we can track which receptor was on before and which is on after, right? So we are trying to get in the BSL-3 facility and do these experiments, but yeah.